Hello everyone, welcome to my channel Anupma Biology Classes. In this video, our topic is light reaction, which is the first part of the mechanism of photosynthesis. This video is taken from Plant Physiology series CBSE class 11, part 3 of chapter 14. Now, let's see here the lecture overview. So in this video, we will focus on some topics like mechanism of photosynthesis, light reaction in which non-cyclic electron transport pathway, cyclic electron transport pathway, chemiosmotic hypothesis to explain photophosphorylation, difference between non-cyclic and cyclic electron transport pathway. Now, let's start the topic in which our first topic is mechanism of photosynthesis. So, the process of photosynthesis completes in two steps, in which the first one is light reaction and the second is dark reaction. Light reaction is also called photochemical phase and occurs in thylakoids, whereas dark reaction is called thermochemical phase and occurs inside the stroma of chloroplast. So, photosynthesis has two parts. First one is the light reactions, where light needed to produce organic energy molecules, that is ATP and NADPH. And the second is dark reactions, in which no light needed. Instead, dark reactions use ATP and NADPH to produce energy molecules. So, the main differences between the light and dark stages are: in light stage, which is occur in the grana, while the dark reaction occurs in the stroma. Light reaction is light dependent, while the dark stage is light independent. In light stage, ATP produced, while ATP used up in the dark stage. In light stage, NADPH produced, while NADPH used in dark stage. Oxygen is produced in light stage, while the carbon dioxide is used up in the dark stage. In light stage, chlorophyll molecule is involved, while in case of dark stage, chlorophyll not involved. So these are the main differences between the light reaction and the dark reaction of mechanism of photosynthesis. Now, the first step of photosynthesis is light reaction. So what is light reaction? Light reaction begins as soon as light falls on green plants. The pigment molecules present in photosystem first and photosystem second absorb their specific wavelengths of light and become energized. The accessory pigments absorb light and transfer their absorbed energy finally to their reaction centers, which take part in the photochemical act. The two photosystems are connected in series with each other by the components of electron transport chain. The reaction centers become so excited that they escape high energy electrons which move to nearby electron acceptor molecules. The electron moves through two pathways. First one is the non-cyclic and the second is cyclic. The non-cyclic electron transport involves participation of both photosystem second and photosystem first, whereas cyclic electron transport involves only photosystem first. Now, in light reaction, first part is non-cyclic electron transport pathway. So, here in this picture, you can see that a schematic diagram of the electron transport pathway in the photosynthesis involving two pigment systems. The light energy, in case of non-cyclic electron transport pathway, the light energy of the specific wavelength is absorbed by the chlorophylls and the accessory pigments of the photosystem second. These pigments transfer their absorbed energy to photosystem second reaction center, which is P680 or chlorophyll A680. The reaction centers P680 becomes photo-excited and exudes an electron with a gain of energy. The electron of photoexcited P680 is taken by pheophytene and transferred to quinone. Once the quinone gets reduced, the P680 by losing an electron becomes oxidized, that is, gets positively charged. The oxidized P680 must get an electron to return to the ground state for the next photochemical act. Now, in case of green plants, the required electrons comes from the water by the process called photooxidation of water or photolysis of water. This process occurs in oxygen evolving complex located in the lumen side of the thylakoid membrane. It is responsible for the splitting or oxidation of water and evolution of molecular oxygen. It is catalyzed by an enzyme called water oxidase. Oxygen evolving complex requires presence of manganese ions, chloride ions and calcium ions. Evolution of one molecule of oxygen requires breakdown of two molecules of water which release 4 hydrogen and 4 electrons. Thus, the electron released from water are immediately accepted by oxidized reaction center of photosystem second. It is believed that electrons are accepted by photosystem second reaction center through an unknown substance, that is jet, and hydrogen ions temporarily stay in the thylakoid space, that is the locules. The high energy electrons that leaves photosystem second, here you can see that photosystem second, that is P680. So the high energy electrons that leaves photosystem second are captured by quinone, which is the primary acceptor, which send them to an electron transport system consisting of plastoquinone, that is PQ, here you can see that, then cytochrome complex and plastocyanine. Every time electrons passes from donor to acceptor, the reduced donor is oxidized and the acceptor is reduced. For example, reduced quinone gets oxidized by transferring its electron to the plastoquinone and the latter gets reduced. The reduced plastoquinone transfers its electron to cytochrome complex, which finally transfers to plastocyanine. The electrons of plastocyanine are picked by the photosystem first. Now, after this process, simultaneously the pigment molecules of photosystem first complex absorb solar radiation and transfer their absorbed electron excitation to photosystem first reaction center, that is P700, here you can see. So the P700 gets excited and exudes an electron which goes to reduce an electron acceptor A. The oxidized reaction center of photosystem first takes electron from plastocyanine and comes to the ground state. Now, the electron emitted from the P700 is accepted by an unknown acceptor A, which transfers its electron to ferredoxin, an iron-containing protein, which is positioned at the outer surface of the thylakoid membrane. The reduced ferredoxin donates its electron to NADP+. The NADP+ takes electron from ferredoxin, protons from the medium and gets reduced to NADPH in presence of enzyme ferredoxin NADP reductase. So, this is all about the non-cyclic electron transport pathway. So, in this case, in light-dependent non-cyclic electron flow, in photosystem second, that is the P680, photons excite chlorophyll E to an acceptor. Electrons are replaced by the splitting of H2O, that is release of oxygen. Electrons travel to photosystem first down an electron transport chain. As electrons fall, ADP converted into ATP, that is photophosphorylation by chemiosmosis. Then photosystem first, P700, fallen electron replace excited electron to primary electron acceptor. Second, ETC, that is electron transport chain, transfers electron to NADP+ to NADPH. These photosystems produce equal amount of ATP and NADPH. So this is all about the non-cyclic electron flow. Here you can see that. So in a process which is called non-cyclic photophosphorylation, electrons are removed from water and passes through photosystem second and photosystem first before ending up in NADPH. This process requires light to be absorbed twice, once in each photosystem, and it makes ATP. So here you can see that the process of non-cyclic photophosphorylation, and where the chemiosmosis occur, when the enzyme to make ATP, where the photolysis occur. So this is all about the non-cyclic, and in this case the products of non-cyclic electron flow are ATP and NADPH, and it occur in the thylakoid membrane. Now, after this, the second part of the light reaction, which is the cyclic electron transport pathway. So, what is the cyclic electron transport pathway? Cyclic electron transport occurs only occasionally when synthesis of carbohydrate is curtailed due to limited supply of carbon dioxide and NADPH starts accumulating. At this time, there would be no need for additional NADPH. Moreover, the cyclic electron transport serves the purpose of more production of ATP when needed by the chloroplast. The cyclic electron transport pathway begins after the photosystem first pigment complex absorbs light. The pigment transfer their energy to photosystem first reaction center, that is P700. The outer valence electron of the excited P700 is raised to higher energy level, which is captured by the primary acceptor of photosystem first. The primary acceptor A then transfers electron to ferredoxin. Reduced ferredoxin, unable to reduce NADP plus, return the electron to photosystem first via cytochrome B6, plastoquinone, cytochrome F, and plastocyanine. So the electron transport is called cyclic because the electron emitted from the photosystem first returns back to photosystem first, passing through several intermediate carriers. So this is about the cyclic electron transport pathway. After this, the chemiosmotic hypothesis to explain photophosphorylation, that means light reaction, in which both are included, that is non-cyclic photophosphorylation and cyclic photophosphorylation. So the photosynthetic phosphorylation or photophosphorylation is the process of phosphate group transfer into ADP to synthesize energy rich ATP molecules, making use of light as external energy. The mechanism of ATP synthesis in photosynthesis has been explained by Michel in 1961, called chemiosmotic hypothesis. According to this hypothesis, the ATP synthesis is linked to development of a proton gradient across thylakoid membrane. The actual site of ATP synthesis is ATPase complex located on the non-appressed portion of the thylakoid membrane, particularly towards margin. So here you can see in this picture F0 and F1, which is the part of ATP synthase. During photosynthesis, synthetic electron transport, hydrogen protons accumulate in the thylakoid space. Here you can see hydrogen protons are released into thylakoid space after each split of water molecules during photooxidation and after each electron transport between PQH2 to cytochrome F. Increase in the number of hydrogen protons in the inner space of the thylakoid causes the pH of one side of membrane to decrease. At the same time, the pH of thylakoid membrane towards its stroma side increased due to utilization of hydrogen during conversion of PQ to PQH2 and at the time of reduction of NADP+. This difference in the hydrogen concentration on the two sides of the thylakoid membrane leads to the building of a transmembrane proton concentration gradient. This electrochemical proton gradient of protons, that is high hydrogen ion inside thylakoid and low hydrogen ion outside in stroma, acts as the driving force between the conversion of ADP and PI into ATP. This process is catalyzed by ATPase located in the thylakoid membrane. So, what is the structure of the ATP synthase? Since thylakoid membrane itself is not permeable to proton, they move towards stroma side of the membrane through ATPase complex. The ATPase enzyme complex consists of two parts. Here you can see that coupling factor 0, that means F0, and coupling factor 1, that is F1. The F0 is embedded in the membrane and forms a transmembrane channel to facilitate diffusion of protons across the thylakoid membrane. The F1 remains protruded towards the outer of the membrane. It is this protruded portion of the ATPase complex where ATP is synthesized. So, downflow of protons from high to low concentration along hydrogen ion concentration gradient through ATPase complex provides the energy that allows an ATP synthase enzyme to produce ATP from ADP plus inorganic phosphate. It is now well established that the single molecule of ATP is formed from every 3 hydrogen ion passing through ATPase complex. So this is about the chemiosmotic hypothesis. Now, what are here you can see that the assimilatory power. The light reaction generates assimilatory power in the form of ATP and NADPH, which is used in the synthesis of carbohydrates in the dark reaction. Now, if we talk about the concept of cyclic and non-cyclic photophosphorylation, earlier, it was believed that the electron transport and ATP production are coupled, where energy released in electron transport is trapped in the synthesis of ATP. The ATP production during non-cyclic electron transport was termed as non-cyclic photophosphorylation, and the ATP production during cyclic electron transport was termed as cyclic photophosphorylation. However, this hypothesis of chemical coupling has now been replaced by chemiosmotic hypothesis, which is proposed by Peter Michel in 1961. According to this hypothesis, photophosphorylation occurs at the thylakoid membrane when hydrogen protons move from thylakoid locule to the stroma through ATPase complex due to pH gradient across the membrane. It is believed that the single molecule of ATP is formed from ADP and PI when 3 hydrogen ion passes through the ATPase complex. Therefore, the photophosphorylation is called non-cyclic when ATP are formed because of proton coming from non-cyclic electron transport system, and it is called cyclic when ATP are formed as a result of proton coming from the cyclic electron transport system. So here you can see that the schematic diagram showing the involvement of pigment protein complex and ATPase complex in the photophosphorylation. Now, after this, what are the main differences between the non-cyclic and cyclic photophosphorylation? So at first, in case of non-cyclic, photosystem first and photosystem second are involved. Electron travels back to photosystem first. No photolysis of water. No oxygen is released. No NADPH is produced. While in case of the cyclic photophosphorylation, electrons return to the photosystem first by their incorporation to cytochrome. Only photosystem first is involved. No photolysis of water, no oxygen is released, no NADPH is produced. So the main differences are, in case of cyclic photophosphorylation, photosystem first only involved. while in case of non-cyclic, photosystem first and photosystem second both are involved. In cyclic, reaction center is P700, while in case of non-cyclic, reaction center is P680. In case of cyclic, electrons released are cycled back, while in case of non-cyclic, electrons released are not cycled back. In case of cyclic, photolysis of water does not take place, while in case of non-cyclic, photolysis of water takes place. In case of cyclic, only ATP synthesized, while in case of non-cyclic, ATP and NADPH plus H plus are synthesized. In case of cyclic, photophosphorylation takes place at two places, while in case of non-cyclic, phosphorylation takes place at only one place. In case of cyclic, it does not require an external electron donor, while in case of non-cyclic, requires external electron donor like H2O or H2S. In case of cyclic, it is not sensitive to dichlorodimethylurea, while non-cyclic, it is sensitive to dichlorodimethylurea and inhibits electron flow. So these are the main differences between the cyclic photophosphorylation and non-cyclic photophosphorylation. So this is the end of video in which we knew about light reactions and their steps. The next upcoming video in this plant physiology series will be on the dark reaction or biosynthetic phase. So if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.